Well, it looks like today the profile, tomorrow maybe some very important treatments for cancer and cardiovascular disease. So nice work, great work being done. I'm sure we're going to have some follow-up questions for Brian, but first we want to hear from a couple of additional speakers, also talking about nanomedical structures and devices which are in development today. And again, Brian, thank you for just being so right on point on that. It's really good. Our next speaker will be Alice Prokop, and Professor Prokop holds a Master of Science degree in Chemical Engineering from the Institute of Chemical Technology in Prague, as well as a PhD from the Institute of Microbiology from the Czechoslovak Academy of Sciences, also in Prague. Dr. Prokop is currently a research professor at Vanderbilt University, and during the past years of his graduate career, of his postgraduate career, he has published over 160 papers and reviews in the fields of interest which are on point today. Dr. Prokop's current interests involve bioartificial organs and target cancer therapy, creating, I think, a very nice segue from Dr. Helmke's previous presentation. Dr. Prokop, welcome.
Thanks for the kind introduction and also for your invitation to participate in this conference. So, where do we start? Next. Okay. My talk is a little bit simple because of the previous presentation by Russell this morning. Obviously, I don't have to define all the terms. I'm going to address two issues, although I believe the second issue is more important, at least to my heart, and you will see that it is setting new stage for biomedicine development. So the first topic talks about basically assembling new nanocarriers for drug and gene delivery, cellular, intracellular accumulation, and retention by means of these nanoparticles. Differential localization and targeting means will be discussed, and future applications. The second part, which is short, will briefly review the existing modeling tools and approaches to quantitatively describe the behavior of targeted nanovehicles within the vascular and tumor compartments, an area of particular importance. In addition, I will consider elementary strategies related to the complexity to tumor or off-tumor delivery. We'll also stress the importance of multi-scale modeling, that is from the bottom up. That is the area of systems biology, so we can understand the whole problem dynamics, the whole system dynamics. This new discipline is called computational systems biology. Now, part one deals with the nano-delivery technology I've been developing for past 10 years. It represents a unique method to deliver medication by targeted and controlled release of drugs with a possibility of intracellular drug uptake. Nanoparticles are made from all kinds of natural polymers. They are mostly of grass origin. What is important is size and charge. We'll touch upon that. And here is a little bit about how the nanoparticles are assembled. They are made of oppositely charged polymers. They assemble almost instantly in about 3 milliseconds of time and form structures which are of colloidal nature. Now, because of the variety of polymeric solutions used, usually 2 on one side and 2 on the other side, 2 cations, 2 anions, the resulting vehicle is extremely stable in the bloodstream. This is the key to the in vivo application. Charge is also of importance. Usually cationically charged formulations are more desirable, but other charges are as well. That means anionic or neutral are equally good. Size, charge, and stability data. As I said, the stability is of extreme importance. Of course, one starts with stability in water and then serum stability as a model system for in vivo blood applications. We are mostly talking about in vitro injection, IV injection of these nanoparticles, although topical injection is also feasible, particularly for antigen delivery, which I am not mentioning at all in this talk. Freeze drying has been also mentioned as a very important step in preserving the properties of nanoparticles and their reconstitution upon the bringing nanoparticles into physiologic media. Now, of course, the loading of nanoparticles with some important drugs is of importance, or for delivery and slow delivery. And of course, there are some definitions here. Let's go further. Of course, these numbers I'm listing here for entrapment are not very high. They still have to be optimized and improved. But essentially is that the macromolecules are very easily entrapped. Small molecules have to be conjugated to the constitutive polymers of nanoparticles. Here are examples. Now we distinguish between high molecular weight carriers, which are the top 2 lines, high molecular weight alginate and cellulose sulfate. But lately we have been using low molecular species on the anionic side, chondroitin sulfate, low molecular weight alginate. Both are cleared by kidneys if the particle is dissociated. And of course, cations are also very small species. They are listed there. And small molecular weight allows very condensed state of that nanoparticle assembly and also guarantees stability. This is a little bit of drawing how the batch processing is done. We also have continuous process, and typically we produce 200 nanometer size particles, but lately close to 100 nanometers. That's of importance for certain uptake phenomena and avoiding non-specific uptake with the macrophages and REST system. Here is very brief depiction of the effect of pH on charge and size. And you can see that the size is relatively flat for low molecular weight species on the right picture, regardless of pH. This is of extreme importance because nanoparticles typically aggregate around the neutral pH. Example of the morphology, it's not very nice picture like Russell presented for his lipid-based nanoparticles, but that's the case. We cannot improve it. Now, the size is of importance for uptake, intracellular uptake. I'm only going to be very short here. There are several mechanisms of uptake. Usually 2, 3 are functional for given nanoparticle chemistry. What is important is that there is some interaction at the surface between the environment and cell, then perhaps carrier-enabled uptake, It's not specific receptor-mediated uptake, but resulting is intracellular appearance of nanoparticles inside a cell. Now, in many cases, we want to avoid endosomes, that is digestive organelles, and end up in cytoplasm. This is the best route. And some of these mechanisms listed on the left allow that. Of course, in our case is gene delivery to nucleus. Nanoparticles of our provenience provide both, depending on the charge. Well, I have to say that at this moment, the knowledge of the intracellular trafficking is very poor. This is general statement about other technologies. So how to control particular localization within the cell is still highly questionable matter. Zeta potential, that is the charge, is also of importance. I mentioned aggregation. And this is the, again, repetition on that processing with little more detail. So the one solution is introduced through the oscillating tip, and that mist is captured in cationic solution. Now, lately, we can avoid the ultrasonic device and just mix two streams, and that process is easily scalable. So interim conclusion, nanoparticles are of grass origin mostly. Some of the constituent polymers are similar to amino acids, like PMCG, which is similar to arginine. We can adjust the size to less than 100 nanometers. We can avoid this time RES interaction, non-specific interaction. Charge is of importance, and the process is scalable. Let's move to uptake and targeting. So that can be improved by using targeting motifs. I'll show that. And this is feasible because the periphery of nanoparticles is loaded with free amino acids, free primary amino groups, and they can be functionalized. This is example of FACS flow cytometry. Localization on the surface on the left, internal localization, you see fluorescence is lower, and there is confocal microscopy in support of this. I'm going to skip this. We have spent a great deal of time to look into mechanism of uptake. It's really important for further development of this technology in terms of FDA approval, etc. But non-functionalized nanoparticles interact non-specifically. They are not carried inside the cell by some specific receptor. Here is some example of low affinity targeting. Anyway, two motifs can be used to enhance this low affinity targeting. So in this case, there is a protein or peptide derived from thrombospondin. It's labeled TSP521, important macromolecule in the vascular physiology. And this peptide is coupled covalently to PMCG terminal amino group, primary amino group, by simple covalent reaction. Another method uses PEG presentation. That means polyethylene glycol as very flexible ligand can be covalently attached to that targeting peptide and then present it on the surface of the nanoparticles by simply co-entrapment method during the processing of the nanoparticles. So this compound is added either into the internal or external solution and then appears to be on the surface and allows for targeting. high affinity targeting, we tested RGD motif of 3 amino acids, which is actively involved in upregulated, or it serves as a ligand for upregulated integrin at cancer. This is example of higher fluorescent uptake with high affinity cyclic RGD motif. The red peak here, and compared to different controls. So this RGD allows for better affinity to vascular integrins. What is also essential is that the nanoparticles, once they are taken up, are retained. They are not effluxed. They are not ejected from the cellular environment. This is just one experiment showing that the relatively stable fluorescent signal is there over a period of 300 minutes. These nanoparticles are not targeted. Okay, I will move further. Time is short. So the control release of entrapped protein is of importance for slow and controlled delivery over a longer period of time. So we have used 2 methods, either entrapment or covalent, actually 3 methods, covalent attachment of certain protein entity to constitutive polymers, or non-covalent sheaf-based complex, which can dissociate upon the disturbances in the environment of the cells, like pH or ionic strengths and so on. It's clear that the non-shift, non-covalent attachment via shift base is a very potent method because it allows certain drugs to be available in free form where they are active. Now, this is an example of slow release and how we can control it with some cross-linking of the particle. So we sew the particle by means of polydextran or polyaldehyde compound, and we can slow down the release almost to nothing. Okay, this is a release over time. Next is non-cross-linked case of fibroblast growth factor, which is very potent angiogenic factor, and release over time, over 22 days, and pretty linear fashion of release, which is in opposite to many different release technologies which show burst effect at the beginning, lots of huge peak of release of a given substance. We also demonstrated the activity of these released compounds in vitro experiments, so they are retained as biologically active molecules. I will skip this. Yes. So we can conjugate in 2 different ways, small molecular weight drugs. I haven't shown too much of that. Doxorubicin is one case and allow for release over a period of time. But what is essential is that the nanoparticles allow intracellular accumulation of that doxorubicin and very high effect and therapeutic efficiency. This is just quick show on the histological observations, tests for different organs, and control. I think control is up. Doesn't matter. There is not much difference. Okay. So now some in vivo data very quickly. Rationale is to target to tumor vessels, blood vessels, and reduce the blood flow to the tumor to suffocate that tumor. We have used basically the difference between markers and tumor and normal endothelium as a means for targeting. Tumor endothelial cells are easily accessible via IV injection. So this is a cartoon, upregulated receptors on the internal wall of the blood capillary, and nanovehicle, which is provided as a ligand for targeting. So now some of the in vivo data. This is example of gene delivery, actually, by using reporter gene expression of luciferase, actually. And this is done in a sponge model of cancer, which is highly neovascularized form of cancer kind of model system. And that virus is adenovirus, and free virus does not express luciferase very much. It's on the right, that circle. But please note the logarithmic scale on the left. So there is about several times larger expression for passive distribution. That means non-targeted particles, but those which are targeted show about 10 times higher ratio to the background. So this is quite successful in vivo case in animals. Then this is example of combination radiation and delivery. We are testing doxorubicin delivery in conjunction with radiation and some other effects. There is a molecule which is tentatively identified as T-cell surface antigen CD5 as endothelial receptor, which is upregulated by radiation. So this is then used to target ligand, which has been specifically fished out by phage display method. It's HVGGSSV peptide. So we modify the nanoparticles with this peptide, show affinity to blood tumor capillary cells, this dense area, and then show in vivo specificity. However, there is a problem with renal elimination, and so the best is to harvest organs. There is overlap between the tumor and kidney accumulation. And by harvesting organs, we show that there is higher binding, 3.2 times greater on irradiated tumors as compared to non-irradiated tumors, using that peptide and pegylated delivery. There is another example of slow release and in vivo application. In this case, cytokines have been entrapped in nanoparticles, actually 4 of them. And in this specific case, 2 allow for tumor reduction in size. That is the GM, CSF, and interleukin-2 entrapped in nanoparticles. The tumor size is considerably smaller compared to the other controls. This is this is for intratumoral injection in this case. Now, there is a shift in T-cell spectrum towards the TH phenotype, which is the desired status. There is also some imaging done, of course. We have already listed some of them. So here is the example of harvesting organs and measuring fluorescence and then plotting the data. Let's go on. So I'll just summarize here very, very briefly that this method allows for very high versatility. Actually, I have presented only 2 chemistries, and many different combinations of polymers can be used. We can entrap different molecules, but in contrast to Russell's technology, We just deliver water-soluble substances, nothing else, okay? So each technology has its own merit. Manufacturing is simple. No organic solvent residues, which are typical for many nanotechnologies. There is strong patent position. Okay, this is this cartoon for this nano delivery technology. We can entrap inside in the core different agents, imaging agent, adenoviral delivery gene, vector. We can entrap drug, often coupled to some constitutive polymer, and we can functionalize the periphery. These positive signs can be functionalized, or through the PEG-coupled moiety. Okay, let's shift to the last section. Because of the time, I will read it rather quickly. So there is a growing recognition in both academia and industry that the prevailing trial and error design of drug delivery techniques is a serious limiting factor. Then mathematical modeling has been suggested as an important tool in the design of drug delivery protocols. These issues include a rational design of appropriate agent strategies for their optimal application and technologies for spatial and temporal control. Systems biology provides methods for computational search and improvement, and it's actually interdisciplinary science. We try to develop some sort of systems biology model for cancer coupled with delivery. What is important here is identification of elementary events at each level of hierarchy. I'm talking about subcellular or molecular, subcellular, that means organelle, cellular level, tissue level, and whole organism level. They have vastly different time constants, and that's important at that computing. Now, this is the definition of systems biology, but let's be short. So we have to identify emergent phenomena at each level of hierarchy. That is the critical set of physical description of each level, which actually reflects the dynamic behavior of that level. So there is lots of genomic, proteomic, and other data available to biologists, but most of them don't know how to handle that data. Systems biology can do that. I will have to only present you with this cartoon, and I'll stop. So this is identification of emergent phenomena at the subcellular level. So we have 3 different signaling pathways which are organized vertically. They have own receptor and own ligand. Each leads to different outcome: proliferation, apoptosis, differentiation, invasiveness, and cancer. There is a difference. It could be highly invasive cancer and sort of benign cancer. Now, biologists mostly look at each signaling pathway independently of others. They don't have the tools to look at the interactions. That is what is called crosstalk between the signaling pathways. Systems biology tools allow to quantify in very detail each individual pathway and then introduce and test different crosstalks computationally, and also interrogate these final schematics, that is putting stops or upregulate or downregulate genes or reactions at certain levels to avoid cancer. So this is going to be future of biomedicine. I'm not going to be critical of this conference. Of course, there were some systems approaches mentioned here, like ethical issues. But other issues are equally important, proprietary issues, regulatory issues. So they belong to the category what I call soft science. They are not clear-cut. They are undergoing discussions, what is to be required. And failure to look at the problem from the system's view, and this was just a small example of that, can cause that the drug will never show up on the market. That's where I will stop. Sorry for extending.